Hey, how's it going, everybody? Mr. Binder here. Taking a look at Chapter 6, DNA replication. A very important process that needs to occur inside of a cell before a cell divides via mitosis and meiosis. All right, and the key molecule that we're going to be discussing throughout these slides is DNA, or deoxyribonucleic acid. Now remember from Chapter 1, it consists of a deoxyribose sugar, so it's actually missing a hydroxyl group there on the number 2 carbon of the sugar. That's why it's called a deoxyribose sugar. This is it still a ribose sugar, but because it's missing a hydroxyl group on the number 2 carbon, it's called a deoxyribose sugar. Very important in a DNA molecule. Again, it has a phosphate group and one of 4 nitrogenous bases, A, T, C, and G. Now remember, T always pairs with A, and A always pairs with T. C always pairs with G, and G always pairs with C. Now there's some hydrogen bonding that occurs between these two molecules that brings this molecule together, and which is why it has its double helical structure. These are known as hydrogen bonds. And they're just interactions between the molecules, and it'll bring it together and obviously form its shape. All right, remember there's 3 hydrogen bonds that form between C and G, and there's 2 hydrogen bonds that form between T and A. Remember, DNA is organized. We will talk about the organization of DNA later. Right now, our DNA is organized in 46 chromosomes, 23 pairs of chromosomes, 22 autosomal chromosomes, and 1 pair of sex chromosomes. Our entire DNA is about 3 billion base pairs. That's a lot. And across those 3 billion base pairs, there's approximately 20,000, we could say 20,000 genes. And what genes are, they're just codes, okay, sequences of DNA that will specifically make a protein. Okay, so when we say genes, we're actually kind of talking about a recipe book here. And that recipe book is found along our DNA, specific sequences that code for protein. And we'll be discussing that in Chapter 7. Now, taking a look here, there's 4 requirements for DNA to be genetic material. Okay? It must carry information. And what's that information? All right, that information is the specific recipe for a specific protein. Okay? We know that DNA replicates. DNA will replicate inside of our cells. We know this because every one of our cells has DNA. Must allow for information to change. We will be discussing mutation later on, and must govern the expression of the phenotype. Okay, so gene function. How does a gene function? And then how is it expressed? So if we take a look at DNA replication, the process, okay, of duplication of the entire genome prior to cell division. So if we take a look here, this is the cell cycle. This whole thing here, we have different phases. All right, we know that a cell goes through its own life cycle, and it will divide at some point. Now we know that G1, S phase, G2 phase are all the different phases of interphase, and then when a cell is ready to divide, it'll go through PMAT, prophase, metaphase, anaphase, and telophase. All right. G1 phase is basically the growth phase, or the initial growth phase after mitosis has occurred. The S phase is very important here. Okay? The S phase is where a cell will replicate DNA prior to cell division. So in eukaryotes, or in eukaryotic cells, DNA replication will occur during the S phase of the cell cycle. There's a couple rules here of replication that I would like to talk about. It's semi-conservative. What does semi-conservative mean? Well, if we were to take a look at the structure of DNA, we see that it is made of 2 strands. Well, instead of just the whole entire strand getting copied and we have a brand new one, each parent strand will act as a template for a new daughter strand. So at the end of DNA replication, you'll have 2 two DNA molecules. Each of those DNA molecules will have a parent strand and a daughter strand. It's known as semi-conservative replication. Replication will start at origins, specific origins found along our DNA where replication will actually occur. So it doesn't start at one end and zip it all the way through to the other end like a zipper. It'll start at these specific origin sites along the DNA. The new daughter strand will always be synthesized 5' to 3' direction. Okay, so the parent strand actually will be, the parent strand won't be the 3' to 5', but the new daughter strand will be synthesized in a 5' to 3' direction. Okay, and we'll take a look at some issues, obviously, that our cell will have to kind of figure out and provide a solution for it, and we will talk about that. It's semi-discontinuous. It is a solution for anti-parallel strands. And the last thing here, RNA primers are required for DNA replication to start. We will discuss that as well. This page here, okay, this picture is very, very important. This picture will be on the test. You will need to label each of these different structures on the test. Now, I would like you to remember all of these different structures. Okay, so we can take a look at three different things here when we're discussing the mechanism of DNA replication. First, we need the strand to separate. The double helical strand needs to separate. Once the strand separates, we need to build complementary strands. Complementary strands meaning that A always pairs with T, T always pairs with A, C pairing with G, and G pairing with C. Okay? That's what complementary means. And then finally, dealing with errors during replication. Unfortunately, there are some errors. We might have an A pairing with C, but our cell is going to double check. And when it finds that, it'll remove the C or remove the A and replace it with the proper nitrogenous base. Taking a look here, this is the specific pathway that our cells will take when forming a new DNA strand. Taking a look at the different colors here, if the parent DNA strand here is blue or turquoise, after a cell's first successful replication of DNA, each parent strand will act as a template for the new complementary strand to be replicated. And after the replication is successful, again we'll see that each DNA molecule will have a parent strand and a daughter strand. It's known as semi-conservative. And if we were to take a look at, let's say, conservative and dispersive, the reason why our cells will undergo DNA replication using a semi-conservative model is you might get errors in the conservative. And how does a cell know if those are errors if both strands are the daughter strands? This way, our cells are almost kind of keeping the integrity of our DNA strand in terms of what needs to be there in a specific sequence of nitrogenous bases. So we first take a look at strand separation. The enzyme here, we're going to talk about a lot of different enzymes. These enzymes are responsible for separating the strand, adding primers, and then actually building the complementary base, the bases and the complementary strand or the daughter strand of the new molecule. It all starts with separating the strand, and the key enzyme here would be helicase. So helicase binds to these origin sites, which is found along the DNA molecule, and will start to create what's known as replication bubbles. And they look something like that. There does become an issue. There are some not really problems, but challenges that a cell will have to face when replicating its own DNA. There's two things here. The creation of a supercoil. And that just creates tension. As helicase unwinds the molecule, it'll create some tension within the molecule. And the enzyme here responsible for relieving that tension would be your topoisomerase. And what this does, if we take a look at this picture here, it will grab the coil and it will create these relief cuts along the DNA strand. Because it makes these relief cuts, the tension will then obviously dissipate or decrease, and the strand can now continue to be separated. Now, the other problem is the hydrogen bonding that occurs between the nitrogenous bases. If you have A pairing with T and C pairing with G, when helicase comes and separates those strands, or those hydrogen bonds that occur between the bases, they are actually going to want to come back together. All right, remember, hydrogen bonds are just they're just an interaction between the molecule. They're not permanent bonds. They're just an interaction. So if we take a look at that, all right, what our cell will do, instead of those hydrogen bonds forming back together once helicase will separate them, our cell will pop on these proteins, these single-stranded binding proteins. They're almost like caps, and those caps will fit on those nitrogenous bases, okay, preventing the two strands that have been separated from coming back together or annealing back together. Again, we take a look at the site or the specific origins of DNA replication. It doesn't start from one end and head to the other. There's multiple origin spots on the DNA molecule. And the reason why there's multiple origin spots or sites is to decrease the overall time of DNA replication. It only takes about an hour for our DNA to be replicated. So helicase unzips the molecule, okay? Topoisomerase relieves the tension. Now when single-stranded binding proteins are added, okay, to the DNA to prevent it from annealing back together. Well, how do we start this whole process, or how do our cells start this whole process of building complementary strands? Well, it all begins with a primer, okay, a primer. And the enzyme responsible for building this primer would be your RNA primase. You might have heard it as DNA primase as well. RNA primase, DNA primase, these are examples of an RNA polymerase enzyme. But taking a look here, we'll call it RNA primase for now. In the picture it does say DNA primase. We can change that to an R so to not create confusion. Again, it's the same enzyme. It just belongs to the class of the RNA polymerase. Now, what this RNA primase will do is to start DNA replication, will build a complementary strand, or a small segment of a complementary strand of an RNA, okay, RNA segment. And the enzyme responsible for this would be RNA primase. You know, it kind of reminds me like if you want to prime a pump, like a water pump, what do? You fill that pump up with water. Okay, so what we have to do is kind of start this whole process with a primer. All right. Once the primer is added to the strand, DNA polymerase 3 will then attach to the primer and start building the new complementary strand. The molecules that are used here are nucleoside triphosphates, or better known as DNTPs. Okay. The D meaning deoxyribose, the sugar that is found in the DNA molecule. Nucleoside triphosphates. There's three phosphate groups found on these nucleosides, and the reason why there is that those phosphates will be taking part in this whole reaction that's taking place to assemble this new complementary strand. Again, remember, the strands are always synthesized or assembled from 5' to 3'. There's two different strands here that we can talk about, the leading strand and the lagging strand. And this is something that we need to discuss, because DNA polymerase can only synthesize the new complementary strand from 5' to 3'. So it's following the parent strand as a template. in the 3' to 5' direction, but the new strand is getting built 5' to 3'. Again, DNA is an anti-parallel molecule. So this will create an issue when it's building a strand away from the fork, and we'll talk about that. That strand is known as the lagging strand. Now taking a look here, building complementary strands. All right. Again, we'll talk about the leading strand and the lagging strand. All right. This picture here on this side, this is known as the leading strand. RNA primase added the RNA primer at the end here, or at this specific origin. And DNA polymerase 3 attached to the primer and is now building a complementary strand towards the replication fork. In the 5' to 3' direction, the new strand is synthesized that way. The lagging strand, okay, as helicase, and this is helicase here, as helicase unwinds the strand, okay, we can see that the leading strand is built towards the replication fork. Well, the lagging strand, we see here that the parent strand is actually, or the new strand is synthesized in the exact opposite direction, or away from the replication fork. The reason why this is that the new strand is always synthesized in the 5' to 3' direction. Because DNA is an anti-parallel molecule, it cannot copy or cannot create or synthesize the new strand both towards the replication fork. So this obviously creates some problems, but the solution that our cells have is this semi-discontinuous solution that we'll talk about. Again, the leading strand is very straightforward. This RNA primer is added here, okay, by RNA primase. DNA polymerase 3 will attach and build or synthesize the new DNA strand towards the replication fork, and that's towards helicase unzipping this parent molecule. Again, the issue that we'll see is the lagging strand synthesis. And there has to be a couple things that come into play here. Notice the lagging strand, and you might have to compare and contrast the leading strand to the lagging strand. There's multiple RNA primers. All right, because the lagging strand is synthesized away from the replication fork, it has to build it almost in sections. Okay, our cell needs to synthesize this new strand, or the lagging strand, which will be synthesized away from the replication fork, in fragments. And these fragments that are going to be used to build the specific strand is known as Okazaki fragments. About 1,000 to 2,000 base pairs long, and we see that they will be attached kind of later. It's almost like a puzzle piece for the lagging strand. Okay. Multiple RNA primers are used here, multiple RNA primers. So on the leading strand we only saw one primer. On the lagging strand we actually see multiple primers being used here. So we have multiple spots for DNA polymerase to attach to start synthesizing that strand. Okay. DNA polymerase 1. This is an important enzyme because it will remove the RNA primer. Once the RNA primer is removed, again, this is RNA. All right, it needs to be removed in order for the complementary strand to be synthesized correctly. Once that RNA primer is removed, a DNA nucleoside triphosphate will then replace, and that is DNA polymerase 1. It'll fill in the gaps. Once the gaps have been filled in, thanks to DNA poly 1, we see a ligase here that will build the phosphodiester bond, or the backbone. It will kind of put the Okazaki fragments together. Okay? And this is kind of like the last step in terms of forming the complementary strand, okay, on the lagging strand. Again, there's some issues here with dealing with errors. As DNA is replicated, there might be a mistake, or again, we can see A pairing with T, or C, sorry, or we can see G pairing with T. This occurs very fast, so errors can occur. Again, there's about 1 million errors, all right, that occur during the replication of DNA. So it's up to the cell's enzymes to discover these errors and make the correct corrections. DNA polymerase III, as it synthesizes the strand, will again check for errors and make those specific corrections. DNA polymerase II here, this will repair damage after the strands have been synthesized. So it'll kind of double check as once the strand is actually completely synthesized. Now this is just a summary of DNA replication. Again, I want you to take a look at the specific steps here. This is the summary of it. I would like you to review most of these, or if not all of these slides, multiple times. You will have a test question, and it will be matching. You'll have to match the function of each of these enzymes, okay, to their name. All right, so what does DNA polymerase III do? Well, DNA polymerase III will synthesize the new strand once it attaches to the primer. Okay, and then we can talk about primase. All right, what does primase do? Well, primase builds a small RNA primer on the parent fork, and it serves as the initial attachment for DNA polymerase III. These are the functions I'd like you to know, again, as a matching question. The one issue that our cells have is, and this is a phenomena that kind of occurs as our DNA is continuously replicated. It's called chromosome erosion. Now, on the leading strand, okay, there is a small RNA primer that was built, okay, to start DNA replication. Well, on the leading strand, that RNA primer actually is not replaced with DNA, with deoxynucleoside triphosphates. That RNA primer is actually it's kind of just removes itself, and at that spot there, there are, well, basically kind of just erodes, and it's called chromosome erosion. And it happens at the leading strand. Okay, it happens on the leading strand. DNA poly 1 can't replace the final RNA primer on the leading strand here. Okay, as we see DNA poly 1 doing in the lagging strand. All right, and this will create a problem because after every successful DNA replication, we see a little bit of DNA actually shortening here, and it will create a problem. Because as our cells divide, our DNA is getting shorter and shorter and shorter. Now there is obviously a solution to this. It's called telomeres. Okay? And telomeres are repeating non-coding sequences at the end of chromosomes, almost like a shoelace. Okay, a shoelace having a nice little plastic end. It's the solution here, or I guess kind of the temporary solution for protecting our DNA. As we see our DNA, there's a lot of non-coding sequences along our DNA, but there's also, again, specific coding sequences known as genes, and these genes are very, very important for specific cell function. Now, we see here that these telomeres are kind of like a protective cap, but as we wear our shoes out, we see that that plastic end of the shoelace will eventually kind of, fall off. And that's what happens to our DNA as we get older. All right, and our DNA, and that could obviously create problems, in the long run. We see the specific enzyme here that's extending our telomeres, known as telomerase. Telomerase will extend these telomeres. More specifically, as we are developing as an embryo in our adult stage, these telomerases, these enzymes, kind of slow down a bit, which is why the chromosome erosion does obviously occur. And this has everything to do with, it's called the Hayflick limit. So the theory is that our cells can only divide a specific number of times. because the telomeres, these non-coding sequences, will begin to decrease as our cells divide, again, the protection for our genes will be lost, and it can create problems. And what these problems are, they're age-related problems. Diseases, aging, so dementia, atherosclerosis, macular degeneration, cancer, and it's an issue that researchers and scientists are trying to solve right now. Because if you take a look at it, it's pretty simple. If we can extend these telomeres, how long can we actually live? And that's a good question that again, scientists and researchers are asking themselves. I would like you to go through each of the slides, and I would like you to review the whole process of DNA replication. All right, understand what the different enzymes that are involved here, their specific function, and again, review it as many times as you need. Come to class, and we will have a nice little review. There will be a test question on telomeres, okay, on the actual test. I'd like you guys to know this as well. The last section here, again, is the packing of eukaryotic DNA. Okay? Basically, when our cells are done replicating, all right, and ready for division, there needs to be some organization here. Well, we take a look at the organization of our DNA. It's 46 chromosomes, okay? But in order for our DNA to condense into a chromosome, it needs some specific proteins here. These proteins are called histones. They're almost like a sewing wheel, okay, where the thread or spool, a specific spool of thread, that spool is like this histone here, and that thread is like our DNA. It wraps itself around it. Okay. When we see 8 clusters of histones, that's known as a nucleosome. And when we see some nucleosomes or coiled strings of nucleosomes coming together to condense and continuing to condense into our chromosomes, those nucleosomes are known as solenoids. All right, this is how our cells will pack and organize our DNA.